In chapter 4, we're going to be looking at metabolisms, enzymes, DNA. So, what if I asked you, how do you define metabolism? What is metabolism? Stop and answer that question for me for a moment. I bet you probably said it has something to do with how your body loses weight or gains weight. That's what we usually think of when we talk about it. So most people describe metabolism relating to the breakdown of food. But it's not just the breakdown of food, but it's also the building up of components in our body. So metabolism is the sum of an organism's chemical reactions, all of them put together, right? And so we have two types of these. We have catabolic reactions and anabolic reactions. Catabolic reactions, I want you to think of the word catastrophe, a catastrophe. This is when you break down a bigger molecule. You release energy in the form of heat and maybe ATP and then discard what you don't need. So you are breaking down complex molecules into simpler compounds, and you think catastrophe, like you're tearing something out, everything's falling apart, so we're breaking down. An example of this is when you have digestive enzymes breaking down food and then releasing that energy for your body to use. Next, we have anabolic pathways. These consume energy. Anabolism or anabolic reactions, you want to think of assembly. We said catabolic, catastrophe, anabolic, you think assembly. You're putting something together. It's the building up of a larger molecule, something more complex from simpler ones. If you're going to build something, that takes energy, right? Think about if you have children, or if you are friends with somebody that has small children and it's Christmas Eve and they bought a bicycle or something and it has to be put together. And so they're putting this together. It takes a lot of energy. You got to stay up all night long and put this thing together, okay? So you're putting something together. It is the building up, okay? An example of this is amino acids being linked together with a peptide bond to form a protein, a muscle protein. We need ATP. We consume it in the body to make large things like cells, lipids, or proteins. ATP is the energy currency for the body. ATP, that is energy currency, okay? So it's real important that we get straight the difference between these catabolic and anabolic reactions here. So what's really important, the most important points here is catabolic breaks down. If you are tearing something apart, you know, there's those people where they're real frustrated or mad, and I don't really condone this, and I'm not this way, but they go and tear something apart and they just feel better. They're releasing energy, right? Anabolic pathways, you're building something and it takes energy to build. So those are the most important parts. Catabolic, release energy, break down. Anabolic, you need energy because you're building something up, okay? So you want to really be sure that you are able to keep those two straight. We have something called free energy that I'm going to introduce. If you've taken a full university or general chemistry level course, you might know what free energy is. And it's basically just energy available to do work. And we have what we call an endergonic reaction. Endo means inside, so endergonic. And then we have an exergonic reaction, or exo, you think exit, and so out. And so an endergonic reaction is going to absorb energy. We could also say endothermic if we're talking about chemistry. Exothermic is going to release, so exergonic is going to release. The only difference between those two words is one is referring more to the cell and one is more of a chemistry word that we use, but it's basically the same thing. And so with reactions, we can have what we call spontaneity. A spontaneous reaction does not mean the same thing as the connotation of the word spontaneous that we use, but it means will it go forward, will it happen without any energy? So an example of a spontaneous reaction would be the melting of ice if you set it out of the freezer. It is going to spontaneously melt. It's not going to need any extra energy to do so. And if you were to take that water and want it to be frozen, though, energy has to be put in. There has to be a change. So that would be a non-spontaneous reaction. Another spontaneous reaction would be like the rusting of iron. It's going to occur without putting energy in. This is a good summary slide of pretty much what I've said. We've got anabolism. Remember, that is assembly. And then catabolism, think catastrophe. Okay. Let's look at this with a reaction that actually occurs, and we're going to learn about two more types of reactions here. So remember, anabolic assembly. This is where we are assembling or synthesizing, putting together molecules. The process is going to need some kinetic energy in order to position those molecules in a way so that we can actually create a bond between monomers. And you can see that in this picture up here. We have two individual molecules here. We have this one here and this one here, and these now over here have a bond. So this is an endergonic reaction because it absorbs energy. Endergonic. And that means it is absorb energy. Energy is going in, ender. So you have to have some energy in the environment for this to occur. So this is a great example of energy coupling where we use two different processes united by a common energy. And so basically what we do is we use one process to drive the other process to be able to happen. And so this is a dehydration synthesis reaction. We already said that synthesis is assembly we're putting together. So what is dehydration? What does hydrate mean? Hydrate means water, correct? So if you dehydrate, you are losing water. So we are going to put these together by removing a water molecule. Okay, and so you can look over here on the right. We have taken, actually we'll start on the left. You have, let's do a different color here. You have an OH on one side and an H on the other. And so we put those together over here and we make a water molecule. So we pull out a water molecule, and then we allow these to bind together. This has another name that is called a condensation reaction, because if you are producing water, it's kind of the same thing as condensation, right? And so dehydration, synthesis, condensation reaction. You want to be sure to understand that and know that this is the one where you are removing water, you're pulling out water to put something together. So it goes hand in hand with anabolic reactions. So we're still looking at anabolic reaction here, and these are some more examples. These are some that occur in the body. Formation of a fat is here at the top. So we have a glycerol on the left over here, and then we have 3 fatty acid molecules. They are reacting, yielding and making a triglyceride, which is a fat, in a dehydration synthesis reaction. So if we were looking at this, how could we tell? Well, first off, if it is dehydration, what does dehydrate mean? Removing the water molecule, right? And so dehydration, we can see we have 3 water molecules. From these 3 bonds right here, we're pulling off a hydrogen and a hydroxide on each of those fatty acids, and it's putting them together to make this one triglyceride. On the bottom, we also have a dehydration synthesis anabolic reaction. We have a peptide bond, and that is the bond that holds together proteins in adjacent amino acids. This is how we build proteins. We've got 2 amino acids joined with an OH or hydroxide from one side, and then we are pulling a hydrogen from the amino acid on the right, and that forms the peptide bond. You can see that right here in the picture as well. And what do we get also? Water. So you're forming a water molecule. You're removing the elements that you need in order to form a water molecule. So you will always have water on the right side of the equation. The amino acids join by a bond between the carbon and the nitrogen atom. We call that a peptide bond. Okay, so when you remove the oxygen and hydrogen on one side of the carboxyl group over here, and the hydrogen off here, it creates an attraction between the carbon and nitrogen, and that's where they're forming the nitrogen the peptide bond, right there. This can be about 100 amino acids joined together to form a peptide, which is how we make a protein, which is very, very important. Use those in the body. All right, so what does catabolism, what are you supposed to think of? Catastrophe. And what is it? It's the breaking down of larger molecules into smaller ones. And we have another term for this one as well, and we call this hydrolysis, or hydrolysis, as some say. Hydrolysis. I say it that way because we've got two words here that have meaning. Hydro means water, and lysis means loosen. So in an anabolic reaction, we looked at pulling out a water molecule to join something together. So this is pretty much the opposite. We're going to add a water molecule in order to break it down. Now, I want you to look at this picture and realize this is the exact same picture we were just looking at. But if you remember from previous lessons, we've got a double arrow here. What does it mean if we have a double arrow? What does it mean about that reaction? Yeah, it means that it is reversible. So we're going to be looking at this reaction now going this direction. So basically it's flipped. And you could flip this around, but they put it all as a reversible reaction, as they left it going one direction. Okay. So catabolism breaks the larger molecules down into smaller ones. And it does so by hydrolysis. This process releases potential energy. Remember, in anabolic, we put it in. Catabolic, we're releasing energy. So it releases the potential energy found in those chemical bonds. It's exergonic because it releases heat energy. So this is exergonic. Sorry, this pen does not always work great. So it's exergonic because it's going to release energy, often in the form of heat, to the environment. Remember, catastrophe is breaking things down. This is like digestion in your body, or the breakdown of glucose in the cytoplasm of the mitochondria. Hydrolysis, remember, is water loosening. Water is added to split down or break a molecule. So if we are going from right to left here, we are adding water that's going to break that bond, and it's going to let those two monomers be able to just stand alone. So again, these pictures are identical. So remember, we are going now in the reverse reaction, reverse direction. We're going this way now, all right? So we're going to do hydrolysis, water loosening. We are going to add water to a fat molecule, which is going to break the bonds, okay? And you're going to wind up with a glycerol and 3 fatty acids. On the bottom, again, if we're going from right to left, we're going to add water, and that's going to break the peptide bond, and then we're going to have 2 single amino acids left. So hydrolysis can be used to break down carbohydrates, lipids, proteins. A water molecule needs to be used. You need one water molecule for every bond that breaks. Okay, and so at the top there were 3 bonds we needed to break, so we had to have 3 water molecules. If you have a disaccharide, the bond between the 2 sugars breaks, and the water is going to give a hydrogen atom to one sugar and a hydroxide to another sugar, and then you're left with the 2 monosaccharides. So these are opposites of each other, hydrolysis and dehydration synthesis, just like anabolic and catabolic. Hydrolysis now is important. It does need specific enzymes. Both anabolic and catabolic reactions need to be controlled. We need to be able to not waste any energy. We need to not waste any types of molecules that we have to make. We also want the reactions to occur at a rate that meets the needs of our energy utilizing reactions. Remember, we talked about having coupling, and so you don't want to make energy and then have it be wasted or lost as heat. You want to be very efficient. A factory needs to run efficient to save money, while our bodies need to run very efficiently in order to save energy. That's very important. Cells could be destroyed if we waste our energy and we don't have enough energy. So let's look at how do we control all these metabolic reactions. This is something that we don't really think about that is going on in our bodies all of the time. Different types of cells conduct special metabolic processes. However, all cells perform certain reactions like breaking down carbs, lipids, proteins, or nucleic acids. And these reactions take hundreds of specific steps that have to occur in the right sequence. So we have a lot of regulation that goes on in the body. The rate of these reactions is going to be controlled by enzymes. Enzymes are so very important. Metabolic reactions require energy to proceed, and so we have to regulate that, and we also have to regulate that rate. Okay. Quiz time. So what is a metabolic reaction? Or I'm sorry, what is metabolism? Well, if you said the sum of all chemical reactions, you are correct. So enzymes control the rate of those metabolic reactions. They are very important in our metabolism. They can act as what we call a catalyst, which can act to lower the energy needed to start a reaction, and that's what we call activation energy. So let's think about the route or steps we take to get somewhere is a pathway. Well, we have metabolic pathways, and they have to have enzymes control their weight all along the way, their rate. So the steps that you take to have a chemical reaction in the cell is a metabolic pathway. We're going to look at that. Enzymes, remember, these are large proteins. They act as a biologic catalyst. I said a moment ago, a catalyst, let me write that down for you. A catalyst is something that speeds up a reaction without being used up. It's recyclable, okay? It does so by lowering the activation energy. You could say taking an alternative pathway or changing the reaction intermediate. All of those things are the same as lowering the activation energy. Enzymes work in these pathways to speed things up and move reactions forward and keep them going at appropriate rates. So let's look at this diagram here. We've got energy on the y-axis and reaction coordinate. That just means, think about just the time it takes for the reaction to occur would be a good way to say that. And so we can tell a lot of things by looking at this diagram. First off, always stop and study the diagrams in your book. Read the little footnotes about them because they are really very, very helpful if you utilize them. If you don't utilize them, they're not helpful. So we're looking here and we can see we have reactants on the left, which they should be, and products on the right. Well, which one of those has more energy? The one on the left or the one on the right? The reactants or the products? So the reactants are higher, so they have more energy, and the products are lower. So that tells us whether or not we are releasing energy or absorbing energy. Which do you think it is? Answer really quickly. Well, we're releasing energy with this reaction, okay? Because the products have less energy than the reactants. So if we're releasing energy, is it anabolic or catabolic? Yep, it's catabolic, right? It's catabolic and it's exergonic, okay? So just a little review there. So now, even though this is an exergonic or energy releasing reaction, you have to have a way to get it started. It's nice and warm right now outside, and we've been grilling a lot. When we go out and we put the charcoal in the grill, we put the lighter fluid on, is that chemical reaction just going to occur by itself? No, it needs a spark. It needs some way to get it started. And so our metabolic reactions also need a way to get it started. Okay, so that's what we call activation energy. All right, and so what this is showing us here is that without any enzyme assistance, we have to have this much energy way, way, way up here in order to get this started. That is a lot. And so if we find an enzyme that we can use, then that cuts it in, I'd say, almost more than half. That is a big difference in energy to get this reaction started. Now, the end result is the same, but it takes a lot less energy to get it started. And so that's what this is showing us, okay? That it takes less energy to get started. It's important to remember that this enzyme is recyclable. It's not used up. That's important because if it wasn't recyclable or reusable, we would have to keep making more enzymes and we wouldn't be saving any energy, right? That would just be kind of going in circles. So we're going to look at enzymes a little bit. Enzymes have what we call on them an active site. And that's the part of an enzyme that's going to combine with the substrate, and that's basically what we're wanting to work on. Okay. You can recognize an enzyme by the ending of its name. What is that? Does anybody remember? An enzyme always end in a S-E, like catalase, peroxidase, maltase, fructase, something like that. Here is a look at how enzymes work, the action of enzymes. So we have an enzyme here on the bottom, Okay, this is a large protein molecule. It has an active site. This is outlined right here. And then these are the substrates. That can be 1, 2, 3, 10. Substrate basically is whatever you're wanting to work on. It can be something that you were going to join and put together, which is what type of reaction? Yep, assembly. Or it can be something that you want to break down. And what's the name for that type of reaction? Catabolic. All right. And so these are going to control that rate of metabolic reaction, lower that energy that's needed to start that reaction. Most enzymes are globular proteins with a very specific shape. And you've probably already tired of hearing me saying it, but shape determines function. This shape is going to match perfectly with the substrate or substrates, okay? It's going to match where it's going to work on that one particular thing. And then the enzyme can be reused. It's not changed after, okay? And so enzymes are substrate specific, meaning there's a specific enzyme for the specific substrates, okay? And the shape of that active site is how they fit together, how they recognize each other. It's like a puzzle piece. When you have two puzzle pieces fitting together, you can see if you look up here at the top that these fit together very well. All right. Now, sometimes the enzyme will kind of have a reaction where it moves a little close to each other. If we get into real good details about enzymes, you look at that type of thing. I think we do get into a little more detail with that in microbiology. Remember, enzymes are biologic catalysts, okay? And the substrate is what we want to work on. So this is a nice little diagram showing you how enzymes work, all right? The other one we were looking at was an anabolic reaction. We were putting something together. I didn't really point that out, but if you go back and look at it, you'll see we put something together, I believe, anyway. And so this one, we're actually breaking something down. And so we have the enzyme here on the bottom. You have the active site that is going to fit perfectly with the substrate, okay? And in this example, the substrate is sucrose. That is glucose and a fructose bonded together into a polymer. So we want to have an enzyme that is going to speed up the rate of this reaction to help break these apart. And so the substrate is going to bind. When it binds, we call this the enzyme-substrate complex. And you will see it abbreviated this way. Enzyme-substrate complex. We have to have a different name for every step, seems like, okay? And so they're going to bind the enzyme to the enzyme and form the enzyme-substrate complex. And that is going to facilitate the breaking of the bond, and then you are going to release the products. And here the products are glucose and fructose, and if you'll notice over here, the enzyme remains unchanged. It is recyclable. An important example of this is catalase, the enzyme catalase. The substrate of catalase is hydrogen peroxide. You know what hydrogen peroxide is, right? It's a toxic byproduct, actually, of some cellular reactions, but we use hydrogen peroxide, not so much anymore, but to clean a wound, right? Catalase's only job is to break down or decompose the hydrogen peroxide into something more harmless. So when we actually have this produced in our body as a harmful byproduct, we need to be able to break that down into something harmless. And so the action of the enzyme when we use peroxide to clean a, well, actually, let me back up. We do that in order to prevent cell damage. And this is one of the reasons they don't recommend using hydrogen peroxide anymore, is it can be damaging to the cells. But the action that makes it work is when you use the peroxide to clean your wound, the injured cell actually releases catalase. And when the hydrogen peroxide comes into contact with them, the oxygen bubbles, then it breaks down the hydrogen peroxide. The oxygen bubbles are set free. This creates a foam and removes debris from the wound. So it's really, really pretty neat how that actually works. So the rate of an enzyme depends on 3 different things. We have factors that can affect or increase the rate of enzyme-controlled reactions. And these are going to occur on what we call a metabolic pathway, right? So we have a metabolic pathway You know what a pathway is, right? It's a series of steps you take to get somewhere. And so a metabolic pathway is a series of enzyme-controlled reactions that lead to the formation of a specific product, okay? A specific thing that we want. We want to break something down or we want to build it together, okay? Each new substrate is going to be a product of the previous reaction. So this is really kind of like an assembly line. This is an assembly line. So if we look here, we've got substrate 1, and we have enzyme A over here to help go from substrate 1 to substrate 2. Then to go from substrate 2 to substrate 3, we have another enzyme, so that's another step, and we go so forth with C and D, okay? And so this reaction is important to occur. So remember that enzyme names end in ASE. A lot of times they actually reflect what they work on, which is really handy. Sucrase, for example, works on sucrose. Lactase works on lactose, that type of thing. So it's really very handy. So we do have different factors that can affect the speed or the rate of these reactions, these pathways. Some of those are going to be things like the number of enzyme molecules, the number of substrates, the efficiency or how well the enzyme works. The reaction can occur faster if the concentration of enzyme or substrate increases. That gives us more of a chance of molecules interacting and creating a reaction. Enzyme efficiency varies between different enzymes. Some work faster than others, some can catalyze hundreds, or some can catalyze thousands per second. It really just depends on the enzyme. The rate at which a metabolic pathway functions can be determined partially by the concentration of what we call a regulatory enzyme that catalyzes one of its steps. So there might be a particular step that is highly regulated, and that's where they control the rate of this reaction. And we're often going to call that the rate limiting step, and it's kind of like a relay race. You're only, what is this? How does that saying go? A team is only as strong as its weakest link. Your runners are only as fast as the slowest person in the team, right? If you have a relay team, okay? So if you have a limited concentration of this rate limiting enzyme, which you can see between substrate 1 and substrate 2 here, the enzyme, if there's not enough of it, it may no longer affect the reaction rate. They can also become too saturated when the substrate concentration reaches a certain level, making it ineffective. So in other words, if there isn't enough enzyme to keep up with the number of substrate molecules, so this is important that we have the right ratio. This is called a rate limiting enzyme. At high substrate concentrations, the enzyme limits how fast the reaction can occur. We don't want to make more product that we need, or we are wasting time. We are wasting energy, and for the body, energy is money. These are usually, very often anyway, at the beginning of a series of steps because it's important how much product is made later on in the pathway. You don't want that to accumulate, okay? This is an important control mechanism, an important way to regulate. The product often also inhibits the rate limiting regulatory enzyme as a negative feedback type of mechanism. So I can't remember if that slide is next here or not, but so what will happen is if I have enough of that product built up to the amount that I need, it's going to come back here basically and turn that off. So it's a really nice control mechanism, and that's negative feedback, okay? And if you don't have enough, it decreases in enough of amount that the rate limiting enzyme will work again, okay? So that this prevents waste of resources by making too much product, like I already said, is really important. So what makes up an enzyme? Enzymes are biologic catalysts, remember. Catalysts, you can think, help chemistry happen, very important. We have cofactors and coenzymes that play a part as well. Cofactors are going to be non-protein enzyme helpers, and that's going to be things like minerals that you probably have heard that we need, zinc, iron, that's an Fe, copper. It's kind of like a helper molecule, and it's usually going to be one of these metallic ions. It helps the active site get the right shape to bind to the substrate, and so it basically you will have an enzyme here, And then you will have a cofactor, a coenzyme here that helps form the shape that is needed for the particular substrate. They both have the same job. They just have a little bit different names depending on what they're made from. So a cofactor can be an ion metal or a small organic molecule. Coenzymes are going to be organic molecules. That's the difference. Whether they're metals or organic molecules, they get a different name. So there's really the only difference here. Coenzymes are going to be things like vitamin molecules. So vitamins are organic molecules that we can't make and that we must get from the diet, like vitamin C. Okay. These are important. This is why your vitamins are so important, because your metabolic reactions cannot occur correctly without those vitamins and minerals that we need, without these metallic ions or our organic vitamin molecules. Again, these are recyclable, so they're only needed in small amounts. A really good example of this is coenzyme A. This comes from pantothenic acid, and it's needed for one of the reactions that help cellular respiration, or a part of cellular respiration. And so it's really important. Most enzymes are proteins, right? We've already said that. There are some RNA enzymes, but most of them are proteins. The correct shape, we already said, is needed for an enzyme to fit the substrate and function. And so things that change the shape of a protein can really be a problem for their functioning. Exposure to things like heat, radiation, electricity, certain chemicals, changes in pH can denature or alter the shape of an enzyme. So if you think about that protein structure, remember we have primary, which is the order of amino acids, secondary, which is the alpha helices and the beta pleated sheets, and then tertiary is where these form together with some other particular ones to form that 3D, and I know that's really ugly, globular protein, okay? And so any of these factors can cause the bonds that hold this 3D globular structure of the protein together can denature it. And basically what that means is if you have an alpha helix, well, it uncoils. It's kind of like taking a slinky. I mean, have y'all ever played with slinkies? Some of you young folks might not have, but if you take a slinky, a wire slinky, and you stretch it out, what happens? It's ruined, right? No matter how hard you try to put it back to the same shape, it just never quite works the same way again. And that's how it is for most proteins. And that's what denaturization is. Denaturing is going to alter the shape of an enzyme. Most of the time, it's permanent. Most of the time, it renders that enzyme or that protein useless. Okay. Many enzymes can, at 45 degrees Celsius, start to become inactive, and then almost all of them will denature at 55. Okay, very important. An example of a chemical that can cause it to denature is like cyanide. Cyanide denatures certain respiratory enzymes, and it stops the cell from releasing energy from nutrients. And so that's really a good application there, is how cyanide stops your respiratory system from working. So energy is having the capacity to change something, or the ability to do work. The first law of thermodynamics tells us that energy isn't created or destroyed, it only changes form. Okay, that's important. We have lots of different types or forms of energy: heat, light, sound, electrical, mechanical, chemical. We could probably take a whole lecture just to talk about the different forms, energy, and changing forms of energy. So energy can be changed from one form to another. Cellular respiration, which all our cells do, transforms energy from molecules like glucose and makes that energy available. Free energy gives free energy, remember, for the cells to use to do work. The reactions of cellular respiration occur in a particular order, right? And because it's a metabolic pathway, and they're catalyzed by different enzymes, and those enzymes, if you think about a factory and you are trying to build something, where are you going to have a part? Think about a really big factory. Where are you going to have the parts located? Are you going to have them all the way across the factory, or are you going to have them close by? Well, you're going to have them close by, right? And so those enzymes are actually located in the cytosol of the mitochondria, okay, or on the mitochondrial membrane. Often depends on the particular organism for that, okay? So this energy powers the cell's activities, and all metabolic reactions involve energy in one form or another. So we've been talking about energy, so let's look at what that form of energy actually is. Think back to high school biology. What is ATP? ATP stands for adenosine triphosphate. So there are 3 phosphates on the end. ATP, remember, is the energy currency of the cell. It's kind of like the dollar bill that you use to buy or build things within our cells. So to have ATP, each molecule has 3 parts. You've got adenine, an adenine molecule, a ribose sugar, and 3 phosphates in a chain. And you can see that structure here, okay? So here we have adenine is there. This is the ribose sugar, and there you have 1, 2, 3. And these little wavy lines just mean it's a high energy phosphate molecule, okay? So phosphate is important because phosphorylation, breaking those phosphates off, that's the process of breaking those phosphates off, is how we accomplish that. ATP is the molecule that carries energy in the form the cell can use. We do have other energy carriers, but ATP is by far the most important. When ATP loses one phosphate, when it's broken off, as in this diagram, it becomes ADP. Instead of adenosine triphosphate, it's adenosine diphosphate. ATP can then be remade from another ADP molecule using energy from cellular respiration and adding that phosphate back on. So it's a reversible process, and we call that phosphorylation. And that is a very important way, an important mechanism that our body uses in regulation. And it's also tied to controlling, regulation is controlling these chemical reactions, okay? But it also is a very important way that we provide energy, and that's in those high energy bonds. So metabolic reactions require the chemical energy that's stored in ATP. The form of energy is held in those chemical bonds that links atoms together and is released when these bonds are broken, okay? So we burn glucose in a process called oxidation. When glucose is eaten, it is oxidized, and energy is released for use in cellular metabolism. Oxidation is the process where oxygen combines with another molecule and the removal of a hydrogen or loss of electrons occurs. All right. ATP that is undergoing hydrolysis, remember that is water loosening. So if we're water loosening, we're breaking something apart. That's a catabolic reaction, right? So we have ATP, we're removing a phosphate. So we're breaking it down into something simpler, and we're making that ADP. That has a free energy amount, or a delta G, of negative 13 joules. So it releases about 13 joules, and that's an energy measurement of energy. ADP, going the other way, has the exact amount needed to be put in. So if you're making ATP from ADP, you've got to add or use 13 joules of energy, okay? The energy needed to make this bond comes from the free energy in our food as we break it down. ADP then can be recycled back to ATP and used over and over again, all right? And so this is the end of part 1. We're going to look at cellular respiration in the next part, I believe. And I'm going to put a link of me in the classroom actually diagramming a good simple way to remember all the steps. So I would highly recommend, I'm not going to make it mandatory, but I'm going to highly recommend that you do that and you draw that out and that you memorize that. In the classroom, that's one of our essay questions, and I've not found a great way to do that online yet. But as an online student, you need to be very conscious of taking responsibility for your own learning. And this is one of the most important concepts you really need to understand in our body is cellular respiration, because we can't exist without it. And so I would highly recommend that you take that diagram that I draw and you commit it to memory, because you are going to need that later on, I promise. All right.